please tell us a little bit about your research institute and a bit about the project that you're using our technology to work on?
My name is Tomáš Groušl. I'm an independent researcher from the Institute of Microbiology of the Czech Academy of Sciences. The institute is currently the largest workplace in the Czech Republic, exploring the properties of microorganisms in the scope of basic research. In our group, which is the part of Laboratory of Cell Signaling, we are mostly interested in the mechanisms which regulate synthesis of the proteins in the cell under stress conditions.
Why is it important to study stress conditions?
Because the cells has to cope with the stress conditions on a daily basis, and there is also apparent therapeutic potential because many severe diseases, as a cancer or viral infections, are accompanied by the stress.
How do you stress your cells to study the stress response?
We have several ways how to stress the cells. We use a palette of stress conditions which somehow mimic the stress the cells can encounter. In detail, we use, for example, the heat stress or the chemical stresses which are imposed by certain drugs.
Can you describe your research in a few minutes?
As I said, we are mostly interested in the mechanisms which regulates the synthesis of proteins in the cell during the stress conditions. The protein homeostasis is maintained by several mechanisms which are integrated in the stress response. It includes the synthesis of new proteins, stress-specific proteins, for example, molecular chaperones, also the degradation of unnecessary or potential harmful proteins, and what we are mostly interested in, the regulated sequestration of such proteins. This sequestration of proteins into protein accumulations or granules is believed to be cytoprotective. However, the aberrant formation or persistence of such granules is usually linked to several pathologies, as the cancer or viral infections. The dissolution has apparent therapeutic potential, and we believe that we can somehow help our understanding of such mechanisms, that our results set a basis for new therapies or the modification of the existing ones. We are using a palette of molecular biology and cell biology techniques, including the advanced microscopy approaches, and that we are using the yeast or also the mammalian model systems, and especially in terms of yeast cells, which are quite instrumental in our research, the possibility to use the super resolution techniques as LiveCodim has improved our research.
What are you hoping that your discoveries will add to your field of research?
We hope that our research will push our knowledge about the mechanisms by which these protein accumulations are formed or dissolved, seeking therapeutic potential.
What features of using LiveCodim were most interesting for you and the work that you're doing?
We are working with yeast cells, and we especially benefit from the super resolution modality. In comparison to other super resolution techniques like structural illumination or STED or expansion microscopy, the LiveCodim system does not have specific requirements for the sample. In other words, you do not need super stable or bright fluorophores. It also possesses low phototoxicity, which is especially good for the visualization of living cells, and it's also fast enough to capture the dynamic processes in the living cells.
It looks like in this figure that we're comparing data sets taken on wide field confocal and LiveCodim. Could you tell us a little bit more about what we're seeing here and why it's interesting?
We can see here the localization pattern of two different markers of stress-induced granules, the DCP2 protein and RPG1 protein, under the stress conditions in yeast cells, captured by wide field microscope, confocal microscope, and super resolution LiveCodim based. The graph on the right side shows the fluorescent signal intensities along the dashed lines. And as you can see is that unlike the wide field or confocal microscope, which cannot distinguish the two signals of DCP2 and RPG1 proteins, meaning that the peaks overlap, the LiveCodim-based super resolution revealed that the two peaks are indeed separate.
Would you recommend LiveCodim to other researchers doing similar work?
Definitely I do. I believe that the LiveCodim system is suitable for almost all types of samples or cells.